Okay, so I'm going to start first by thanking Asunta for kindly inviting us different institutions in Mexico to collaborate in the Tropicsafe project. And this presentation, as mentioned already, is on it's on phytoplasmas in alternative host plants in Mexico. So the collaborators in this piece of work sorry, it jumped. The collaborators in this piece of research are presented here. The institutions are Colpo, INIFAP, Univo, and CICI, and the researchers are listed there. So I'm going to show a few slides for background. This you already present. Some other presentations have shown this, but just leave them there for a few seconds for the pictures of symptoms. Then phytoplasmas that are associated with the diseased palms, and they can be transmitted by a Plaxius crudus, as reported by Florida several decades ago. And very important tools for these studies are the molecular tools, as in this case I'm showing nested PCR and how it is done, and finally that we can identify group and subgroup. And in the next slides, I'm going to show you a list of plants. I'm just going to show you the slides so you can see that there are too many. And the meaning of this is that in all these 3 slides, we can find 32 reports out of 36 of 16 SR4 group phytoplasmas. Most of them are on palms other than coconut, and also all of them are in countries, in different countries in America. And finally, there are some which are related to non-palm species. Some of them are, again, related to 16SR4 phytoplasmas. So the view here is to show you that there are several species of palms and other plants that are non-palms that have phytoplasmas. Several of them do not show symptoms. So the phytoplasmas are present everywhere. Well, so when the disease started in our region several years ago, we thought of strategic aspects for research and development, and these are presented here. One of them is to study the mode of action of the phytoplasmas to understand the disease, to develop analytic tools for detection, diagnosis, screen of LY resistant coconut germplasm, which is very important. And then if you have those coconuts identified and selected, then we should be able to multiply them very efficiently and very rapidly. So I'm going to show you then the project results in this area of research of alternate plants. And what we did was to search for suspicious plants in the areas in red, and the states were Yucatan, Tabasco, and Veracruz. And this is the list of the different species that were found and the number of positives that were obtained. And as you can see, the subgroup found, these subgroups found are basically the ones shown here are D and A. One of the cases was very interesting. This was related with coconut. This is a site in the north of Yucatan where the plot contain segregating population, hybrid by hybrid cross, and the very susceptible Atlantic tall variety. And here you can see how the number of dead palms was increasing with time. And what was important here is that most of the Atlantic tall, 93%, was lost, and about half of the plants in the segregating population. Here I'm showing you some pictures of the species that in which phytoplasmas were obtained or identified. These are showing the developing of symptoms in 2 of them. This is another case in Tabasco. This is a case in a place far away from Yucatan and Tabasco. And in this case, it was an outbreak where killed several hundreds of plants. Well, this is just to show you the map of, first of all, where the subgroup D is located. And as you can see, it is very widely distributed in Mexico. It's also present in the States. And the one I should show you that I said it's a heavy outbreak. It is here in the red spot. And this is the most recent place where we have found phytoplasmas, little yellowing cases, and it is related, as shown in this map, with the subgroup D. Now, if we overimpose the other subgroups, then the group A is not so widely distributed, but it is the one that has killed more plants, actually millions of Atlantic tall palms in this region. And there are other 2 subgroups, E, which has been associated with only 2 individuals of insects, and B, with a few coconut palms, and present only in Tabasco, and the E only in Yucatan. Well, with this information, I would like to mention, based on this diagram, that the phytoplasmas can be transmitted from a susceptible plant that is infected by Aplaxus crudus or maybe other insects to other plants that are not infected. But they can, I mean, coconut plants, but they can also be transmitted to other plants which are different species, for instance, Trinax radiata, which is a plant that usually shows symptoms, but not in all the infected plants. And some of them have remission, and the population is only affected very little. So over decades, the populations are still there, but there is a positive detection all of the time. So they are reservoir of phytoplasmas. The same could be thought for weeds, which are very many species present. And as they have reported in some of the presentations already, it is a common finding. Well, so reservoirs are, the idea of reservoirs is very important because, for instance, in the case of Trinax, this is how it is located in the Yucatan Peninsula. Same for Cocotrinax ready, and Sabal will be on the other part where these are not present. So if we take these 3 together, the whole peninsula is covered, at least in the coast, with these kind of plants that are being infected with phytoplasmas, but the populations are still there over decades. So it's a permanent reservoir of phytoplasmas. And the same can be said for other species throughout the country. And of course, this should be the same with other countries and throughout the world. So the conclusions from this research are that as a result of the studies presented, the number of plant species associated with 16SR4 phytoplasmas has been extended to Mexico, as well as the number of states in the country with cases of plants affected by LY. In most cases, the phytoplasmas associated are of the subgroups A and D, and in a few cases, subgroup B. And as I mentioned, also a few cases of E for insects. There was an outbreak in the peninsula of Yucatan. And as I said, this was established with very highly susceptible materials. And most of them were lost. And this is significant because this is the first outbreak observed within 20 years. And I think that the fact that it happened because susceptible material was planted, and it is not happening when resistant materials are planted, shows that the presence of phytoplasmas in reservoir plants is important only if susceptible materials are planted. So this will teach us that it is very important to use resistant coconuts for replanting. But since this could be a conclusion from this type of research, then it is also very important to consider that screening, finding LY resistant materials is extremely important. And if we do it the traditional way, it takes very long, and maybe we're not going to be successful because it depends on circumstances. But if we have molecular tools which can very efficiently and quickly tell us that the plant is resistant or not, then we will be in a much better position. And as I mentioned before, if we have these resistant materials, we can identify more, and we link this with the biotechnology for their propagation, then we may be able to have very useful tools to deal with these diseases. Thanks very much. Sí, okay. Aquí veo que me está dando la opción de